Hello, my name is Zachary Harshman, and this is my science fair project, The Effect of Curcumin Capsaicin and Morinda citrifolia on Tumor Cell Growth in C. elegans. For background research, there's an estimated over 2 million cancer cases as of 2025, and, and there was an estimated 618,120 cancer deaths. However, the overall cancer mortality rate is declining. Current cancer treatment methods include radiation, chemotherapy, targeted therapy, immunotherapy, surgery, phytotherapy, and stem cell therapy. This project focuses on 3 specific phytotherapies, which are curcumin, capsaicin, and Morinda citrifolia, each producing neuroprotective and non-toxic traits. C. elegans are microscopic worms that were used as the dependent variables in this experiment. And they can be used for cancer research instead of cells because they have a simple genome, fast lifespan, observable cells with transparency. And some C. elegans, for example, UNC32 is a specific modified C. elegans which can grow germ lines that represent cancer cells. The review of literature establishes a purpose/gap in this research in which the effect of these specific herbs on cancer and tumors is unknown. So is the goal of this project to fill this gap by introducing curcumin, capsaicin, and Morinda citrifolia to tumor-induced C. elegans so that we can measure the tumor growth rate over time. My hypothesis was that if the extracted herbs are introduced to the UNC32 modified C. elegans, then all 3 will effectively slow the growth rate of the germline clumps. However, I believe that Morinda citrifolia will have the greatest suppression effects because not only has it previously shown anti-cancer properties, but also anti-inflammatory and antitoxic properties. This visual representation basically shows how I cultured, fed, and induced the tumors to the C. elegans by first adding the E. coli, which is basically their food, to the plates containing the worms, and specifically the UNC32 C. elegans had to be incubated in 15 degrees Celsius over the course of 3 days, while the wild type C. elegans only were set at room temperature during this process. When adding the variables, I simply, along with the E. coli, mix the curcumin, capsaicin, and the Morinda citrifolia along with a control and added it with their food, essentially. Now for measuring the growth rate over the course of 5 days, I used ImageJ analysis, which basically outlines the area of the C. elegans and the area of the tumor containing the zones containing the tumors, and gave a percentage of the zones containing the tumors compared to the overall area of the worms. For disposal procedures, I killed the worms by adding 10% bleach, and after 24 hours, they were disposed in biohazard waste. The results show after specifically 3 days or 5 days here, which was the final analysis, shows that there is a significant difference in the average percent of areas containing tumors after 5 days. Through shown here, the average percent change in tumor areas covered from days 3 to 5 showed that both curcumin and Morinda citrifolia decreased the overall tumor containing zones, while the control and capsaicin, the zones grew over time. Through an ANOVA test, it was also shown that there was a significant difference amongst all of the 4 variables. In conclusion, we can say that 2/3 of the herbs, the curcumin and noni plant, Morinda citrifolia, decreased the tumor size, and curcumin had a slight better effect. The wild C. elegans had no tumors and therefore needed no ImageJ analysis, and the control and capsaicin showed to have an increase in tumor size over time. For future research, oncologists can consider curcumin as an effective anti-cancer phytotherapy. These are the references.